Hello everyone, welcome to BioBoost. Today we will cover cell cycle topic which is very useful for competitive exams like CSIR NET. We will study in detail about every stage of the cell cycle. Here it is the detailed diagram of cell cycle. So at the end of a division of cell, daughter cells are formed and these daughter cells are smaller than their parent cells. The DNA in these daughter cells is usually half of the DNA content as their of parents, means their DNA is almost half of their parent cell. Now cells grow in size by synthesizing cytoplasmic material to reach almost 4 times of their original volume. Means the cells 4 times that of their original volume. Now the DNA content which is present, it gets doubled by replication. We know that by replication DNA divides. Cells are now ready to undergo cell division. So in every cell cycle, cells undergo division. Now the growth and division both occur alternatively in a cyclic fashion, and this cycle is we call as cell cycle. Growth and division both takes place alternatively, and because it takes place in a cyclic fashion, it is called as cell cycle. Now it involves two phases. First is the growth phase, that is also called as the interphase, and it is usually the non-dividing phase. And the second is the divisional phase, and it involves the division. So growth phase usually involves G1, G2 and S phases, and division is usually meiosis and mitosis. Starting with the growth phase, that is the interphase. So it is the interval between two successive cell divisions. Here, the cell prepares itself by synthesizing and storing substances which are very essential for cell division. So it is also called as the preparatory phase. Now non-dividing cells remain permanently in this stage. The cells which are not dividing, they remain permanently in this stage. That is the interphase. Biosynthetic activities of the cell are at maximum in this stage. So that's why it is also called as the biosynthetic phase or the metabolic phase. Because cells are undergoing many changes biosynthetic wise and everything is getting prepared here. So it is called as the biosynthetic or metabolic phase. Interphase is further divided into three substages. These substages that we discussed above is the G1, G2 and S. Beginning with the G1 phase, it is also called as the post-mitotic phase because it is the first phase which takes place once the cell has undergo mitosis. Here in this diagram you can see mitosis is there. Just after mitosis, G1 phase is taking place, so it is called as the post-mitotic phase. Here, young daughter cells grow in size by synthesizing cytoplasm. Means the cell is beginning to synthesize its own material, its own cytoplasm. Here, the cell synthesizes and stores enzymes, mRNA, tRNA, ribosomes, proteins, and nitrogenous bases, which are needed further for DNA replication. Just before dividing, what cell needs, it synthesizes and stores in this phase. That's why it's called as the G1 phase. Chromosomes here get extended to form chromatin, and it takes 30 to 40% of the total time of cell cycle. Now, in non-dividing cells, that is the mammalian lymphocytes, cell cycle stops at a point in this phase, which is called as the G0 phase or the quiescent stage. Means the cells which are not supposed to divide, the cell cycle stops and this phase is called as the G0 or the quiescent phase. Now the cells which are dividing, they get ready and now they enter the G1 phase. The cells which are supposed to divide, they enter the G0 phase, G1 phase. Next is the S stage or the synthesis phase. Here DNA replication takes place. In this diagram you can see after the G1 phase, S phase taking place. Here DNA replication takes place. Synthesis of histones which are associated with DNA, they also get synthesized, and it takes almost 30 to 50% of total cell cycle duration. Next is the G2 stage or the pre-mitotic stage. The stage which takes place just before mitosis is the G2 stage. In this diagram you can see just before mitosis, G2 phase is there. Here nucleus has double amount of DNA. In this stage, now the cell has nucleus which has almost the double amount of DNA which it originally had. Nuclear volume also increases due to synthesis of rRNA, mRNA and nuclear RNA. Cells synthesize proteins which are required during cell division. Here synthesis of proteins also takes place which are required further into the cell division. Almost 10 to 20% of the cell cycle duration is invested in G2 stage. If we see dramatically, then almost 50% of the time of cell cycle is given to S phase. Mitosis, G2 and G1 takes the rest 50%. G0 stages, a temporary stage which takes place during the G1 stage. So division phase is the last phase of cell cycle and it is the mitosis or M phase. Cell division occurs during this mitosis now. So next we will go to the mitosis phase, which so mitosis is somatic cell division. Here maintenance of chromosomes, number of chromosomes stays same as of parent cell, and in all cells it occurs except the germ cells where we see meiosis takes place. It also includes interphase stage which includes G1, S and G2 stages. Then comes the karyokinesis which is called as the nuclear division. Now karyokinesis is divided into the following phases. First is prophase. So what happens in prophase is nuclear division of mitosis begins with this stage. Chromatin threads lose water and they start to condense. In this diagram below you can see the chromosomes have started to condense. Chromatin threads coil like a cylindrical spring and become shorter and thicker to form chromosomes. We can visibly see the chromosomes in this stage. Each chromosome already doubled up due to DNA replication during S stage. DNA has been replicated, so each chromosome looks doubled up. Here you can see in S stage DNA has been replicated. Now by the end of prophase, each chromatid splits lengthwise. Each chromatid has been splitted into two chromatids. Now nucleolus and nuclear membrane also start to disappear. Nuclear membrane and nucleolus both they start to disappear. Centrioles divide into two and move towards opposite poles. Here in this late prophase diagram also you can see that centrioles have been divided into two and they start to move towards opposite poles. In the intervening cytoplasm, microtubules orient themselves between daughter centrioles and a spindle called amphiester is formed.
So microtubules start orienting themselves. Microtubules like actin, myosin, we have learned about them. So they start to orient themselves between daughter centrioles, and a spindle is formed which is called amphiester, and by the end of prophase, nucleolus and nuclear membrane completely disappear. Now comes the metaphase state. Here in this diagram also you can see that in this stage each chromosome becomes more compact and short. Chromatids separate except at the centromere that has not divided so far. Means chromatids separate lengthwise but except at the middle centromere they stay connected. Chromosomes migrate towards the equator of the spindle and they arrange such that centromeres are at the equator. Centromeres stay attached to spindle fibers. Here you can see spindle fibers. These are the diagram wise spindle fibers, and it is the equator where chromosomes stay arranged. Next is the anaphase state. In anaphase, centromere of each chromosome divides and daughter chromosomes formed by splitting of sister chromatids. Here the centromere which was present in the middle, it divides and daughter chromosomes are formed by splitting of sister chromatids. Daughter chromosomes move apart and pull towards poles of spindle. They are pulled towards poles of spindle. Here you can see they are moving towards the poles. Chromosomal movement brought about by contraction of spindle fibers. Here, centromeres are pulled towards poles and arms are dragged behind. In this diagram, you can see that arms are towards equator and centromeres are towards poles. Next is telophase. Here, daughter chromosomes reach poles and form two groups, one at each pole. Here, chromosomes uncoil to form chromatin net. Chromatin net is formed in this stage. Nuclear envelope and nucleus both reappear around each group. And by the end of telophase, you can see two daughter nuclei are formed. Here, spindle fibers and astral rays also disappear. You can see in the diagram. Last comes the cytokinesis, where in animal cells a shallow groove appears in the cytoplasm at the equator, while in plant cells a cell plate is formed at the equator of the dividing cell. Both side you can see cell plate and cleavage furrow. So finally, two daughter cells are made in animal cells by cleavage furrow, and plant cell plate is formed at the equator. These 2 points you need to remember. So in short, mitosis ensures growth of cells and organisms. Now, this was all about somatic cells. But what about germ cells? So germ cells divide by a process named meiosis. In this, reduction division occurs in the reproductive cells only at the time of gamete formation. Means only when the cell reproducing, that is the germ cells, they undergo meiosis 1 and meiosis 2. Now, definition wise, it is the reduction division which occurs in the reproductive cells only at the time of gamete formation. In this, four daughter cells are formed from single parental cell, and chromosome number in daughter cells are reduced to half. That is, haploid cells are made from it. From single parental cell, four daughter cells arise and their chromosome number is reduced to half. Now, because the chromosome is reduced to half, so there is difference of number. So it is also called as heterotype or first meiotic or a reduction division also. Just like mitosis, meiosis also have stages like prophase. So prophase 1 for meiosis 1, it is longer in duration as compared to mitosis and profoundly modified into 5 stages. These are the 5 stages. First is leptotein stage. This stage initiates meiosis. Meiotic cell is comparatively large with a large nucleus in this stage. Here you can see centrioles are there. This stage has diploid number of chromosomes and chromosomes here are long and thin and longitudinally single. The chromosome has beaded appearance due to presence of dense granules which are called as chromomeres. Second is the zygotine stage which has esters. In this stage, it commences with movement of chromosomes which is brought about by attraction between chromosomes of each homologous pair. Chromosomes here are in pairs. Because they are same, they are termed as homologous pairs and they are attracted by chromosomes. So they approach each other and become intimately associated to form a bivalent. Here, the main thing is synapses is present in zygotine stage. Next is Pachytene stage. It is stable period in the cell division. Each paired chromosome of a bivalent gets shortened and thickened and is more clearly visible in this stage. Homologous chromosomes start splitting into two sister chromatids by a longitudinal furrow. Here, chromosomes start to split into two sister chromatids by a longitudinal furrow. Next is diplotein stage. Here, splitting of chromosomes and separation of homologous chromosomes initiates. Here, bivalent consists of 4 chromatids. Also, synaptic forces lapse and homologous chromosomes uncoil and they start to separate. Because the separation is incomplete and some points of contact is present, that contact is called chiasmata and it remains. Exchange of chromatid parts between homologs occurs due to breakage and rejoining of weaker chromatids at chiasmata. So this crossing over brings about exchange of hereditary material. Now chiasmata begin to move from the centromere towards the ends of the tetravalent and this is called as the process of terminalization. Once the crossing over has brought exchange of hereditary material, then chiasmata brings about a process which is termed as terminalization. Last is diakinesis stage. Here nuclear membrane and nucleolus disappear. Bivalent separate and chiasmata reach end of the chromosomes and also nuclear spindles start to form again. Next come metaphase 1 after prophase 1. Here movement of the bivalents towards the equator of the spindle takes place. Means the bivalent starts moving towards the equator. Now bivalents orient themselves on the equator so that centromeres lie one on either side equidistant from the equatorial plate. Means centromere here lies either side of the equatorial plate and they are equidistant from each other from the plate. Next come anaphase 1. Here, chromatids separate into pairs and move towards opposite poles of the spindle. In this diagram, you can see that chromatids are starting to move, and also they are in pair. So they start to move towards opposite poles. Two haploid chromosomes formed on either pole. Now, two haploid chromosomes are formed. Separation of tetrads into dyads or a pair of chromatids is called disjunction, and separation and it involves the separation of chromosomes that were brought together during synapses. Last is the telophase 1 stage. In this diagram, you can see that sister chromatids are present and they are separating from their pairs, homologous pairs. Here, nuclear wall forms, chromosomes uncoil, and cytoplasm divide into two haploid daughter cells. Now, meiosis second, that is second division. Interphase state in these daughter cells may or may not be present. So, interphase is optional here. Now comes prophase second of meiosis second. Here in this diagram, you can see in prophase second, nuclear membrane disappears and spindle starts forming. In this diagram, you can see. Next, in metaphase second, chromatids connected only with centrioles and lie parallel to one another. Dyads move towards the center and lie on the equator of the spindle. Here you can see they are lying on the equator. Next, in anaphase second, chromatids separate to form sister chromosomes and they start to move towards the two poles of the spindle. Last, in telophase second, chromosomes uncoil and form two groups. Nuclear membrane and nucleolus reappear and four daughter cells are formed from one cell. In the beginning, we started from one cell, and till now, after meiosis 1 and meiosis 2, four daughter cells are formed. They are haploid. So, lastly, cytokinesis takes place. Here, the division of cell cytoplasm and cleavage of cell into two daughter cells, which occurs in telophase along with formation of daughter nuclei. Cytokinesis also takes place during telophase state. When we begin from prophase to metaphase to anaphase to telophase, then along with anaphase and telophase, cytokinesis starts to take place. So it's an ongoing process. So this was all about cell cycle and how in germ cells and somatic cell division takes place. Many questions come from these subjective type questions, especially from part B, comes from cell cycle. So it is very important later on when we will study about cell cycle regulation also, then these points will become important. Cell cycle regulation involves many checkpoints and kinase, CDK kinase that are very important from CSIR net perspective. So here it was a brief, it was a basis for you all. It is very important and there is no need to go through books to learn this. And if you like this video, please like and share and also subscribe. I will continue to make such videos on CSIR concepts, and thank you so much for watching. I will try to make a summary of cell cycle 2 in my shorts video. Till then, thank you.